This idyllic scene is deceptive. That's because each winter beekeepers can lose some of their bee colonies. In recent years, however, colony losses have been persistently heavy. It's not uncommon for beekeepers to lose a third of their stocks. What it means is that we as practitioners, as beekeepers with 20, 30 bee colonies, have had whole colonies dying on us, and that's the problem. All of a sudden we're faced with empty hives. Yes, that's it. And a few years ago, 8 or 9 years ago actually, I lost half of my bee colonies. That was a really difficult year. Colony collapse disorder was the term used to describe the phenomenon when it hit the headlines. Scientists have been searching for some time for effective treatments to control the parasites which are causing massive bee die-off in a large number of countries around the world. The factors which influence bee health are many and varied, but experts agree Varroa destructor and the infectious diseases spread by this mite continue to represent the biggest threat to European honeybees. The fact is that the Asian Varroa mite is deadly to Western honeybees. In our case, Varroa first appeared around the beginning of the 1980s, but there was very little of it to start with. We didn't take any action. Then later it became more of a problem, and the first losses occurred in the Biebergemünd district. Studies were conducted locally, but they weren't any help. Sometimes there were more Varroa mites around, and sometimes fewer. The situation varied in different colonies. There were also quite a few collapses. But there is one thing on which scientists the world over agree. The biggest killer is the Varroa destructor mite and the microorganisms transmitted by it. Controlling the mite is thus becoming the top priority when it comes to keeping our bees healthy. In Germany alone, the Varroa mite destroyed around 30% of all bee colonies in 2011 and 2012. That's around 300,000 of the million or so bee colonies that exist in Germany, according to Professor Grunewald, head of the research project on the Varroa situation in a selected region of Hesse.
We have
been conducting the Institute of Bee Research field study in the Main-Kinzig district since 2008, so we now have data from 5 years of research. Our idea is to get as many beekeepers in the region as possible on board so we can treat large areas with alternating products. 2/3 of the beekeepers in the region are actually involved now, and we have gathered a lot of useful data over the last 5 years. These show variations in mite infestation at the different sites and even extreme differences within an apiary. Outside the brood cells, the female Varroa mites live on adult bees. They sit on the cuticula, the bee's body armour, and find their way to the soft intersegmental membranes. Here they bore a hole and feed on the bee's blood or hemolymph. This is how they transmit disease pathogens in the same way as ticks. Reproduction takes place solely inside sealed brood cells. Shortly before the cell is capped, the female mite moves in and slips under the developing bee larva. Before it can lay eggs, it must first feed on the hemolymph. If you open up a brood cell, you will therefore find the parent mite at the base of the cell, possibly already with Varroa offspring. Young bees are often damaged before they emerge. Wing deformities are a sign of viral infection, for example. In case of severe infection, larval bees often die before they hatch. The research project on the Varroa situation in the Main-Kinzig district, a selected region of Hesse, started in 2008 and will end in spring 2014. Between 2,500 and 3,000 colonies have been included in this study. 200 representative colonies belonging to 18 different beekeepers were selected for the core study. All the colonies were treated at the same time, and the number of Varroa mites on the bottom board was then documented. The apiaries are spread over the entire district, and both rural and urban sites were included. The numbers of Varroa mites counted per week fluctuate considerably year on year, ranging from just over 200 mites per population on average in 2008 to over 1,000 in 2011. Not until 2012 did the numbers drop back.
3 Ziele haben wir mit dem Feldversuch im Blick.
In our field study, we have set ourselves 3 objectives. First, we asked ourselves whether effective mite treatment leads to high winter survival rates. We started the late summer treatment at the same time throughout the district. The winter survival rates we actually observed over the years were considerably higher than 90%. Our second objective was to gather reliable data on an area over a number of years. Finally, we wanted to look at whether blanket treatment would be effective in reducing mite pressure throughout the district. Effective control of Varroa infestation is crucial to preventing excessive winter losses of bee colonies. The treatments began after the honey harvest, when the bees were being fed for winter. Baverol and checkmite are the products which have been used because they are highly effective, alternating between the two each year in order to prevent the development of resistance. Once the strips were inserted, the mites which dropped onto the bottom board were counted each week. The treatment covers not only the mites actually on the bees at the start of treatment and the mites as they emerge from the brood cells, but also the mites which enter the colonies from outside during the treatment. Let's start by summarizing the main conclusions of the field study. Both Baverol and checkmite are highly effective. No evidence of resistance to the active ingredients was observed. The winter survival rates were considerably higher than 90%, except for 1 year. The mite load in the individual colonies within an apiary is highly variable. The mite drop counts rose steadily in the colonies between 2008 and 2011. The beekeepers involved are very satisfied with the safety and efficacy of the treatment. I have 40 colonies and have been participating in the study for a number of years. The experience has been very good for me, and I have suffered very few losses. I have been treating bees for years now with various products, and I have not experienced any losses so far. For 5 years now, we have been involved in a field study in Oberursel, conducted by Frankfurt University, with various products, or rather Baverol and checkmite strips, alternating between the products. In recent years, I have experienced no losses at all, apart from 4 colonies which were queenless, which is probably normal. Otherwise, I've been very satisfied up to now.
Der Feldversuch hat ein unerwartetes Ergebnis erbracht.
The field study produced one unexpected result which became apparent from the second year of the study. During this long, warm autumn, dead mites continued to be recorded in high numbers on bottom boards in the bee colonies, rather than the gradual decline in numbers which might have been expected. In some cases, the number of these mites actually increased. At this point, we investigated whether this might be due to a decrease in the efficacy of the treatment, but this was not the case. There was therefore only one possible explanation. The mites were being repeatedly reintroduced into the colonies as a result of colony reinfection through contact with neighbouring colonies. We were particularly taken aback by the extent of this phenomenon. In some cases, more mites were reintroduced later than had originally been present in the populations. This made it very clear that around 30% of colonies belonging to other beekeepers not participating in the field study was sufficient to prevent a reduction in infestation over the entire study area.
Die Behandlung mit Baverol und checkmite im Wechsel.
Alternating treatment with Baverol and checkmite is keeping colonies mite-free to the extent that we now lose only 1 to 3% of the colonies over winter. We know that the beekeepers not participating in the field study have losses of 20 to 50%. For us, the focus is on the quality of the honey, which is why I have all my honey tested regularly for residues by the State Institute in Hohenheim. No residues of either Varroa or Checkmate have ever been detected. I must point out, however, that the combs located near the strips during treatment do not go into my honey super. They remain in the brood nest or are used for forming nuclei. Residues of Varroa mite control products in honey can give rise to uncertainty and debate and ultimately damage the image of honey. That's why we need information on the behavior and fate of the active ingredients used in the bee colony. It's our laboratory's job to determine the risk of residues associated with the individual treatment methods, so any preventive action required can be taken in good time. At the same time, we try to offer recommendations to our beekeepers based on our data, which comes from almost 3,000 honeys and 500 wax samples. Beeswax is interesting in this regard, as wax is the only source of residues in honey. If nothing is detectable in the wax, then there should be no residues in the honey. Careful handling of the wax can dramatically reduce the exposure of the bee colony to active ingredients and thus also the risk of measurable residues in the honey. Our beekeepers are now more aware. They recognize and avoid mistakes. We now have better mite control products, and we also use them in a more targeted way. This has also become apparent in the honey and wax samples from beekeepers who have participated in the field study for 5 years. Where beekeepers had a rational wax management program, almost no residues were detectable, either in the honey or in the beeswax. But honey production itself is now under threat because bees' health is in danger, and up to a quarter of bee colonies have been dying over the autumn and winter each year for many years now. Under these circumstances, it's essential to have reliable figures to hand regarding the Varroa mite load. But the field study also showed how important it is to monitor the effectiveness of treatment and also to retreat individual colonies if needed. We would also like to see successes and failures being discussed openly within beekeepers' associations and common approaches developed wherever possible. So our study has produced a lot of new data on the Varroa problem in Germany and shows the major challenge the Varroa mite continues to pose for our bee colonies. We can see, therefore, that Varroa destructor is still one step ahead of us. Varroa-resistant bees and the ultimate method of Varroa control are not yet on the horizon. The risk of treated bees becoming reinfected is underestimated. Continuous monitoring of the effectiveness of treatment and, where possible, the degree of colony infestation is required, along with a flexible and coordinated approach to control, using tried and tested, authorized methods and products, what we call integrated pest management. Long-term Varroa control remains a top priority for science and practical beekeeping, which means that even greater efforts are called for.